Here you go.
JJ, let me know when you want to get going. It's your show. So as Cartier sits, we'll get going. It's my show? All right, we're going to get started. Welcome all you capstone carnivalers, carnivalees, whatever we call them. Today we have two NSSs, and I'm going to NSURS. Today we have two NSURS, and I'm going to introduce the first one, Chloe Smith, who I've had the joy of working with over the last 4 years. But before I do that, I want to introduce a special guest, her mom, in from New Mexico, Tanya, is here with us today. And we would not have it any other way. Is that a squirrel on a stick? Yes. Just noticed that. Well, it was. Chloe has been here all 4 years. She's a ENS double concentration in sustainable agriculture and sustainable forestry. And she's also been a mainstay on the herb crew for a few years. Chloe's one of those special students for me who came in the last time that I taught a first year seminar. And I've had a chance to see Chloe navigate her way through school. She's done an awesome job with that. And I knew Chloe had something special because during that first year seminar, when I asked everybody, told everybody that their first project was going to be dredging a pond, she was one of the first ones in. And she also was my comrade on our ill-fated trip to get a mud pump and try and implement that. We'll leave that go. Chloe's, one thing that really tells me, Chloe's abilities, when I first proposed this project to her, most students would have run away. Chloe ran with it. She did a really great job. I'm not going to give away the project right off the bat, but I'll tell you right now that Chloe ran with this and actually developed one of the most eloquent and elegant projects that I've worked with a student. Small amount of variables, high return on results, and I will leave it at that.
Thanks, Dave. So the title of my NSURS project is Germinability of Multiflora Rose Seeds Ingested by Oats. I'd like to begin by introducing everybody to what an invasive plant species is. A species is considered invasive when it is both non-native and has the potential to cause environmental or economic damage. Often these species tolerate a wide variety of habitats, grow and reproduce rapidly, compete aggressively for resources, and lack natural pests. Environmentally, these species might decrease biodiversity through many ways, but examples of those might be through modifying disturbance regimes, modifying soil conditions, outcompeting native species, or changing plant communities. Economically, the U.S. spent $34.7 billion last year in invasive species control. Multiflora rose is just one of these species. That's especially a problem in Western North Carolina. Multicolor rose was brought to the United States from Japan to act as a living fence along highways and in many reclamation projects. Today, it is considered invasive in 41 states. It grows in dense, thorny, monocultural thickets and is pretty hard to get rid of. Like most invasive species, multicolor rose grows rapidly and outcompetes native species. It does this through spreading through layering, which is when an individual cane will grow roots and form a new plant, and through seed dispersal, that's animal mediated. In Western North Carolina, the primary disperser of multicolor rose is birds, specifically songbirds. Pictured on the right is a typical cluster of multiflora rose hips. Within each one of those hips is 1 to 21 achenes. Achenes are often mistaken for the true seed, but are actually the hard outer seed coat that protects the seeds, as seen in this diagram. The achene is softened naturally through frost, and that triggers embryo growth. Multiple studies have shown that bird digestion aids in softening of the achene and increases the probability of germination. Multiflora rose hips provide one of the main sources of food for birds in the winter. If you can think about all the songbirds that we have in Western North Carolina and the range of all those songbirds, it really makes sense that multiflora rose is a big problem in this area, especially if you consider that one plant can produce up to 500,000 seeds. In many cases, chemical and mechanical control methods are used to get rid of multiflora rose. These might include cutting back the plant, mowing the plant, using a backhoe or a tractor to physically excavate the plant, or there are many toxic herbicides that people use to control the plant. Often, these ways are really time and resource demanding. In some cases, using goats as a biological control method may be a viable alternative to chemical or mechanical control. And for those of you that don't know, biological control is when you use one species that's a natural predator or enemy to control an invasive species. So goats, unlike other livestock, are browsers, which means they're better able to defoliate plants more thoroughly than grazers like sheep or cattle. Additionally, they're resistant to plant toxins and bloating. Multiflora rose is a really thorny, dense bush that we don't really want to touch, but goats love it. And it's really desirable for them because it's high in protein and nutrients. So unlike any mechanical or chemical control, goats can convert weeds into milk or meat, which are potentially profitable enterprises. Rent-to-goat businesses where landowners pay to have goats clear their land of unwanted species are becoming really popular, or more popular, in the US and the Southeast. In the Buncombe County area alone, we have 4 of these types of companies. Using goats seems like a really straightforward answer to getting rid of multiflora rose. However, if these goats are eating multiflora rose during the time that it has its seeds, they may be taking those seeds to other areas, and if the seed remains viable through their ingestion, they might be continuing the problem of invasive species. Endozoochory is a study of animal-mediated seed dispersal. This is really relevant when you're studying multiflora rose because birds are the main dispersers. Therefore, it is important to look at if goat ingestion has the same effects as bird ingestion on germination. This is especially cool if you're a land manager who owns goats, because in an agricultural or goat rental scenario, you may be able to be in complete control of seed dispersal if you plan carefully. I broke my methods, and This brings me to my objective. I wanted to look at whether goat ingestion enhanced or depressed seed germination of multiflora rows. I did this in 3 parts. First, I needed to collect the seeds and then feed them to the goats. I collected the seeds. I collected entire branches containing mature seeds in October of last year at Red Wing Farm in Swannanoa, North Carolina. I fed these seeds to 14 full-grown goats of similar ages and similar weights. I intentionally fed the entire branch to the goats as opposed to just taking off the seeds because I wanted to mimic a real-life housing scenario, just to make sure the goats would actually eat the seed and not just eat the leaves. I then collected the feces 24 hours after I had fed the goats. This was fairly easy because the goats were isolated during the time period. I chose the window of 24 to 48 hours to collect the feces based off the methods of a similar endozoochory goat study that looked at germination after goat ingestion. And I collected a total of 7.3 pounds of feces. I then needed to get the seeds out of the dung. First, I dried all of the dung on a screen in the heated greenhouse for 48 hours. And then, with some help, I picked through every single pellet very carefully to find the seeds. My control seeds were collected from rose hips from the same bushes that I collected at the same time, and I took the seeds very carefully out of the rose hips. I tried really hard with both the treatments not to damage the achene at all. All the seeds were disinfected with 3% hydrogen peroxide. So in total, I found 72 seeds in the dung. Therefore, I used 72 undigested seeds to act as my control. Before I planted them, I needed to mimic the 3-month cold stratification period, so they were in the fridge for that amount of time. Then, with each seed, I placed them in a small petri dish between 4 pieces of filter paper that were kept wet for the entire germination period. Each petri dish was assigned a number, and I randomly placed them on a table in the greenhouse. I monitored all these dishes daily for 6 weeks, and I considered a seed germinated if the radical length was between 1 to 2 millimeters. And when a seed germinated, I took it off the table and counted it. At the end of the 6-week period, 16 of my control seeds germinated, and 3 of my ingested seeds germinated. These values may seem really low. However, multiflora rows and other species within the Rosa genus all show similarly low germination success. So I was sort of expecting to get these low numbers. I used a 2x2 contingency table, which is a chi-square analysis, to determine if there was a significant difference between the 2 proportions. This test resulted in a p-value of less than 0.006, which suggests the ingested seeds germinated significantly less than my control treatment. The results from my study suggest goat digestion depressed multiflora rose seed germination through some mechanical or chemical process within the goat gut. This image shows the physical difference that I observed between the 2 treatments of seeds. As you can see, the ingested seed is way smaller than the undigested seed. This difference in size leads me to believe that the digestive fluids of the goat were able to not only break the achene, but also completely destroy the embryo within the true seed. This hypothesis is supported by 2 similar endozoochory studies that looked at seed germination after goat ingestion. In both of these studies, it was found that prolonged retention in an animal's gut results in decreased germination in seed species with similarly hard seed coats than multiflora rows. The results of these studies might also explain why avian digestion increases germination and goat digestion depresses germination. The diagram here represents rumination in a goat. This means they have 4 compartments in their stomach, and they chew their cud, which means that they regurgitate their food back and forth between their rumen and their mouth to chew it before digestion is complete, as indicated with these red arrows. In contrast, avian digestion is a much shorter process, and it's much less complex. As seen on this right diagram, there's only 3 compartments in avian digestion, and in many songbirds that are consuming these multipolar rose seeds, there's actually only 2 compartments because many songbirds in western North Carolina don't have a gizzard, which physically grinds the food with rocks. So the amount of contact within a bird's digestive system is just enough to scarify the seed coat, whereas the seed is in the goat's digestive system for a much longer time, which is able to destroy the seed embryo. Personally, for me, it was the most important to find the practical implications of my study for goat owners and land managers. So multipolar rows will typically hold onto their seeds from August till February, which is a fairly long time. And normally, if you were going to go in and mow these bushes or cut them, you wouldn't really want to cut them during the time when they have the seed, because it might fall on the ground and contribute to the seed bank or become a new plant. Whereas if goats are completely destroying the seed, then you do want to go put them on your land with multipolar rows during the time that it has the seed on it. Because they may contribute to reducing the amount of multipolar row seeds within the seed bank, which might contribute to long-term management of multipolar rows. This information might also contribute to a more successful year-round goat rental business, because there is no time limitation on when you should use goats on your land for multipolar rows. There's still a lot we don't know about the role goats play as seed dispersers. Future studies that want to look at multipolar rows and goat ingestion might look at the number of seeds that successfully pass through the goat gut as a function of time. An example of one of these studies might collect dung at 12-hour intervals after feeding them to look at, or to just further insight into the effects of gut retention on seed germination. Another study also might want to look at the effect that dung has on seed germination through fertilization. They could do this for multipolar rows or for any other plant species. I would also be interested to look at, or to see future research on other invasive species in goats, such as looking at the relationship between seed size or the thickness of the seed coat and post-gut germination. All of this information could really contribute to a more successful and more well-planned rent-a-goat business model. I would like to thank Dave and my committee, and Christopher and Beth from Red Wing Farm, and all of these people, plus all of these goats in the blue. I really hope everyone without them. Questions?
You said that birds are known to be a major disperser. Have there been studies of birds with multipolar rows and their effect on the seeds? Do you have numbers on germination rates?
I can't think of numbers off the top of my head, but yeah, like specifically, birds are the main disperser of multipolar rows.
And so what was
and it's through ingestion, and they increase the germination of the seeds. Yeah. So their digestion is able to just scarify the seed, because the seed coat imposes dormancy, but ingestion through a bird breaks that dormancy. Yeah, and that's why it increases germination. Mark.
I was struck by that figure that one plant can produce 500,000 seeds or something like that. I'm just trying, because when I looked at the seed, so I thought these must be like almost microscopic seeds, but then when you had it on the filter paper, it was pretty big. So I'm just trying to figure out, some doesn't add up. Is that over the whole life of the plant?
Yeah.
The plant lives for 20 years, or how?
Yeah,
how
I
do you
think
get
it
that
is
figure?
over the entire life of the whole plant. I thought about it at first, and I was like, oh yeah, that's definitely possible, because some plants are like huge, you know? But I honestly think that figure is over the entire life of a plant.
And do they live that?
I actually don't really know how long they live either.
The hydrogen peroxide treatment of the seeds, is that something that you got out of the literature? Or is
Yeah. So most of the time when you take seeds out of like a fruity, like a rose hip, they'll mold. So you use hydrogen peroxide to decrease that mold. I also washed them in like a really diluted bleach solution, and it all came from like a rose growers forum. Because as you can imagine, it was very hard for me to find information on how to grow multiflora rose, which was a huge time-consuming thing. And I ended up finding really old literature from China, like Chinese nurseries, on how to grow multiflora rose from seed. But the hydrogen peroxide treatment came from just like a rose growers association.
So some of the seeds still germinated after they went through goat.
Mm-hmm.
Then presumably if you had a goat in one field, a rental goat, and moved it to a different field the next day, it could poop out the rose seeds
Right,
in a different
exactly.
field and still disperse them.
Right, exactly. It's not the most clean results because some of them did germinate, and that's why I think it would be interesting if looking back, you think about all the things you could have done. But looking back, I wish that I had fed the goats and then collected their dung at different intervals, like at 6 hours and then at 12 hours, and then grown those as like separate variables, because the way that I did it is the goats were just isolated in the barn for this period of time, and I collected that dung from the barn. I had like cleaned it out of all the dung I could see before. So I don't really know.
So my question, I guess, is if you were a rental goat person, how would you keep from dispersing multiflora rose to your plants' fields?
Yeah, that's basically like the whole context for my entire project.
Right. So what would you recommend then?
Oh, I see what you're saying.
Happening.
I guess what I would recommend is that people who own goats and have these rental businesses, that they have a specific paddock that they bring their goats to before they bring their goats to a different piece of land, and that paddock is just for them to poop all these seeds out and let them grow, I guess. So they're not dispersing seeds to like another client's land, but to this specific paddock. And they could do that within the 24 to 48 hour period, because that's when the most seeds are go all the way through the digestive system. Mm-hmm. Ian?
From your herb interest, are the roses also very high in vitamin C, and can you use them in a herb potion or?
Oh yeah, like people use multiflora rose seeds in like teas and various stuff. I'm not at all suggesting that we plant multiflora rose. I guess it's not the worst plant ever.
But they're there.
They're there.
We have time for one more question.
Mm-hmm.
So I moved into a house that actually the previous owner thought they were beautiful, and they have them everywhere.
Mm-hmm.
And so I've not seen the report. I cut them at the wrong time and everything else, and I haven't getting more. So can you save me some time and put me onto goat rental?
Save you some time and money, and you can just ask me later.
Thank you.
Thanks, Lloyd.
Thank you.
Are there any announcements from the faculty?
I have two announcements. I have had a request for the last couple of weeks to announce the Pew Grant winners, and you may or may not, students may or may not know that we have an in-house grant program for undergraduate research in the sciences called the Q Grant that we administer here locally. And this semester, we gave 6 students are going to benefit from those, and those are Andrea LaPlante, Kirsten Moore, Joseph Coleman, Joey Shaw, Jane Martindale, and Colleen. So congratulations again. So every semester, beginning of semester, we'll be announcing another round of Q Grant things for something like if you need more than $100 or so, that's a source for that. And then my other announcement is faculty, in case you all haven't noticed Joe Young's emails, there's a presentation tonight by the current NSURS research students on the progress report of their research, and it's 6 o'clock tonight at, where is it? Where's the March 1? 200. Okay.
Any other announcements?
Support local research. Some of your colleagues some of your fellow students are doing the sex study, which happens every 2 years. It's a student-led, student-created, student-organized study of what's going on campus, and it helps us inform the administration about what they can do to help improve reducing STIs on campus. So please support that at your meal. Just take 5 minutes, go in. It's only 4 pages, real fast. Just check boxes. So support your students. Appreciate it.
Any other announcements? Paul, did you want that other announcement? Did you add another week to the semester?
I'm cutting off.
You got it? All right. All right, it's my pleasure to introduce the second seminar today. Chris Rodriguez. Chris comes to us from Fort Lauderdale, Florida. A big fan of the Miami Heat. We beat our Bobcats, unfortunately. He's a environmental studies major, concentration in sustainable ag. Directly related to that, he's worked on the farm for most of his time here, just about I think all but 1 semester. 3.5 years on the farm. Last year and a half as the cattle crew manager, so that's directly related to the project that you'll see right here. Chris likes living life on the edge, and that includes he likes to skateboard and ride motorcycle. You may have seen the motorcycle up there. In fact, he told me something about motorcycle riding is to take the place when he's too old to skateboard, to get a little excitement in his life. And that extends to his choice of his natural science seminar topic as well. It took him a while. He went through a couple different choices about what he wanted to do with the natural science seminar project. I think he would tell you he wouldn't recommend necessarily doing it the way he did, putting it off. But he finally actually ended up with a great project where it's one of those where it ties in with the work program also, and it was actually sitting right under his nose the whole time without him realizing it. So he actually ended up doing a nice job on this, and it's one of those where hopefully it'll actually be not just research sake, but it'll actually benefit some of the stuff that we do on the farm. So Chris, take it away.
Thanks, Mark. So as Mark said, I've been working on the farm for 3.5 years, and for the past year, my responsibility has been the student cattle manager. In the past couple of years, we introduced a new breed into our cow herd, and just working on the farm, talking with some other crewmates, we noticed one group of calves might have been gaining weight at a faster rate than another group of calves. And that just inspired me to look into the data and do this project. So the title of my project is Rate of Gain and Temperament Analysis of Angus and Angus Hereford Calves on a Warren Wilson College Farm. So let me put my study in the greater context of sustainable agriculture. On the college farm, we produce grass-fed beef. It's a more natural system for the cattle. They utilize the ruminant function to digest forages rather than just consuming concentrates in a feedlot. It's better for the environment. There's less fossil fuel inputs and lower environmental impact overall. Grass-fed beef producers tend to be better stewards of soil and water than feedlot farmers. And the beef is better for us. It contains healthier fats and antioxidant vitamins. There are two key components involved in grass-fed beef production: pasture management and cattle management. Our main tool that we use is rotational grazing. Our beef production system is very hand labor intensive. We're constantly working closely with the cattle, moving them on a daily basis. In contrast to conventional production, very little of our work is mechanized. And I feel like we tend to put a lot of emphasis on the forages that we're planting and cultivating for our cattle, the energy, mineral, and protein content, as well as establishment and persistence. However, the cattle genetics and cattle selection is equally as important, and I think sometimes it gets shadowed by the importance of forage in grass-fed beef production. So, like I said, cattle management is very important. We need our cattle to be able to finish in 16 to 24 months. That means from birth to slaughter, they need to reach 1,000 pounds in 16 to 24 months, and that's concurrent with conventional feedlot finishing. That's for us to be a good model for profitable grass-fed beef production enterprise. The cattle we like on our farm are moderately framed, deep-bodied, easy fleshing cattle, and they're favored for their feed conversion efficiency and high rate of gain. Another important quality of cattle on our farm is that they should have a docile temperament. We're a student-run farm, so oftentimes we have inexperienced students working cattle in potentially dangerous situations. This is a picture of the vet trimming one of the bull's hooves in the corral, and he's very experienced, but he might catch us doing some sketchy stuff like that. And so cattle, and further, excitable cattle can prove to be dangerous in a situation like this. If you have a bull that's just real crazy, he could kick you or squash you, kill you, any number of things. And further, cattle with docile temperaments have shown improved average daily gain, carcass quality, and steak tenderness over those with excitable temperaments. The bottom line, cattle that gain at a high rate and are easy to work with are highly desirable on hormones and culture. So let me introduce one of the breeds that I was working with in this study. Black Angus is the most popular breed in the United States. Historically, we had a very mixed herd. We had Simmental, Santa Ana Botrytis, Devon. We even had some dairy breeds. We had all kinds of breeds in our herd. But 10 years ago, we began an intense Angus breeding program where we bred exclusively with Angus bulls, and we purchased some purebred males as well to obtain a more consistent beef product, as well as raise the quality of the beef we're producing. Today, we have an Angus-based cow herd of 65 head. For those of you that are not familiar with cattle nomenclature, a cow is specifically a female that has had 2 calves. However, we include heifers in this equation as well. So basically, we have 65 breeding females, Black Angus-based breeding females. The other breed I was working with was Hereford. This is the second most popular breed in the United States. And the American Hereford Association promotes this breed as being moderately framed and docile. And they say that they convert feed efficiently and gain at a higher rate with beef quality comparable to Angus. Laura's seen that one like twice already. So in 2012, we introduced the Hereford breed into our Angus-based cow herd by way of a bull. This is not the actual bull that we used, but this bull is related to our bull that we had. He's of the historic Trask bloodline from Anderson, South Carolina. Mr. Trask was a cattle producer that raised Herefords in the 30s and he specifically tried to create a line of Herefords that was profitable on grass, because it was the time of the Great Depression and they weren't feeding a whole lot of grain. So that's where our bull came from. And we bred in the 2012 breeding season with our 3 other Angus bulls as well. So we basically have 1 Hereford bull representing the Hereford breed and 3 Angus bulls representing the Angus breed. It would be ideal to have more Hereford bulls, but we just didn't have that on our farm. And then feedlot studies report higher average daily gain with Angus Hereford Anguses. However, these studies are funded by the American Hereford Association, so there's potential for bias, and they're also feedlot studies. So I wanted to do a grass-based study to see how the Hereford Angus crosses would perform compared to the straight Angus calves in a grass-based system. So the objectives are to determine if the introduction of the Hereford breed into our Angus-based program will increase overall herd average daily gain, and also to determine if this introduction will improve the overall herd temperament. And we did, and I evaluated this by looking at differences between the straight bred Angus calves and the Hereford Angus cross calves. So after the 2013 calving season, we ended up with 28 Angus calves and 16 Angus Hereford calves. My methods. Data was collected from 4 different weigh sessions to obtain the results of this study. During the final weigh session, calves were evaluated for temperament using the method developed by Hall et al. The first measure of temperament we took was catch score. Catch score is based on the disposition and the working chute. I had an assistant who was experienced in cattle working and cattle working behavior, though unaware of the objectives of the study, assigned each calf a score of 1 through 5. So it was a blind study. A score of 1 indicated that the calf was calm, there was no movement. A score of 2 indicated the calf was slightly restless. 3, the calf was squirming, occasionally shaking the chute. 4, there was continuous, very vigorous movement and shaking of the chute. And if the calf was assigned a score of 5, it was rearing, twisting its body, and struggling violently. And I'll mention that this was a routine weigh session, and we didn't do anything out of the ordinary to obtain the temperament values of this study. The second measure of temperament I took was exit time. And exit time is recorded as the time it took a calf to travel 1.8 meters after departing the working chute. We used a stopwatch and a piece of rebar that you can't see. In conventional and bigger studies, they use lasers, but that wasn't in the budget. Burrow et al. Suggests that cattle with more excitable temperaments display faster exit velocities. That is, if the cow is very excited and worked up in the chute, it'll just jump out of the chute rather than trot out at a calm pace. So, average daily gain for the entire length of the study was calculated, and that was the pounds gained by each calf divided by the number of days of the study, which was 168 days. After I obtained that value, I looked more closely and I calculated average daily gain between each of the 4 weigh sessions to see when the actual difference occurred. The mean catch score and mean exit time were calculated between the 2 groups as well, and t-tests were used to identify the differences. And that's the gain right there. So, this was the first measure that I took, and the first calculation I made. And you can see the Angus Hereford crossbred calves display a slight advantage over the Angus calves. The Angus calves gained an average of 1.53 pounds per day, whereas the Angus Hereford calves gained an average of 1.67 pounds per day. And that may not seem like a lot, but it represents a real dollar value advantage. Then when we look more closely, we see that they were gaining at a comparable rate throughout most of the study, but then right at the end, the Angus continued to decline, whereas the Hereford kind of leveled off. And I want to make clear that the cattle are still gaining throughout this whole period. It's normal for them to gain less as it gets colder and the winter progresses, but here the Angus was gaining about 0.3 pounds per day, whereas the Angus Hereford was gaining about 0.9 pounds per day towards the end of winter. And this is a graph isolating that final growth period. And it has significant p-values for the cumulative ADG, which I showed earlier, as well as this. So the next thing I looked at was the temperament. The Angus Hereford crossbred calves displayed a lower catch score overall. They displayed a catch score of 2.4, whereas the Angus calves displayed a catch score of 3.2. Most of our cattle aren't too dangerous or excitable in the shoot, but it was interesting that the Angus Hereford calves displayed a lower catch score, because that was another thing we speculated about. The particular bull that sired those Hereford crossbred calves was very docile. You could practically go up and pet him. So I think he passed some of that on to his progeny. The next measure, exit time, also favored the Angus Hereford crossbred calves. It took them longer to travel across the 1.8 meter section, indicating a slower exit time and a less flighty nature overall. So to sum things up, the Angus Hereford crossbred cattle exhibited higher average daily gains overall, especially during the final spring turnout growth period. The crossbred calves displayed calmer catch behavior as indicated by the lower mean catch score. They also exited the chute at a mean longer interval, indicating a less excitable and flighty disposition. We must be careful not to extrapolate these findings beyond the realistic boundaries of this study. Because we only had one Hereford bull siring all of our crossbred calves, it's possible that these advantages we're seeing are linked more to that bull in particular rather than the Hereford breed as a whole. Ideally, we'd have more bulls so we could just get a better representation of the breed. And in larger studies, they use like 600 cows and 15 bulls of each breed, but we just don't have the cattle to do that here. And data on average daily gains should be collected until harvest of the cattle, because we saw that kind of sharp difference, and it's possible that the Angus cattle could catch up with the crossbred cattle as the spring and summer progresses, but this study had to end here, so. And any questions?
Just wondering about the range of ages within your
They were different ages, but it was an even distribution within each group. Yes.
Wondering what sort of background information you had about the heritability of temperament in cattle. Can you talk a little bit about, would you expect that to be something that would?
I read a study where they were specifically looking at that, and they concluded that temperament is a heritable trait. But I don't have any like hard data or anything off the top of my head.
Okay.
Just with something that last thing you said, how many more months until they are?
Well, the oldest ones will begin being slaughtered in about June.
Okay, so it is soon.
Yeah, so most of the cattle in this study are between 700 and 800 pounds, and they'll be slaughtered around 1,000 pounds. So they're not too far off. Yes.
Sorry, another one. What would you recommend to the farm based on your study?
Well, I would just say that this introduction of the Hereford breed was a good idea. A few years ago, Chase tried to cross our Angus cattle with Devon, and it didn't work out so well. The cattle gained at a slower rate than the rest of the calves, and so I would say this one worked out pretty well. And the Hereford Association people, they say that the real benefit is with the females. So we're going to retain a lot of these crossbred calves as replacement heifers, and they're supposed to have better conception rates and easier calving. So that would be a cool study for the future. Yes.
All right. Which ones taste better?
I'll tell you in a couple months. Bingo.
So, what I understand, the breeding was started at with an Angus quote-unquote base.
Yeah.
Would you expect to find similar results if they were to start with a Hereford base or the
I don't know. I read a little bit about that. Typically, they cross Angus cows with a Hereford bull, just because there's so much more Angus cows. I think so many more.
Time for one more.
Yes, you.
Are most of the crossbreds came out black with, or
Black with
also
a
red?
black with a white face, and then there is one that was red with a white face.
I was wondering whether there was any difference in terms of wintertime growth and color, and some being warmer and not using as much, or using energy to stay warm. Whether there was any.
That would be interesting. But they're pretty much all black with a white face.
Okay.
But they do say that red Angus perform better in summer than black Angus because they're not as hot, they're more active.
That would be interesting.
All right.